Hello, welcome to this week's video lectures. Today we're going to go into Unit 4, where we're going to talk about adaptive immunity and that third line of defense. Remember that first line of defense are those physical and mechanical and genetic barriers that prevent bacteria and pathogens from getting into the body from starting an infection. The second line of defense are those cellular responses to uh, general are those general cellular responses like inflammation, phagocytosis, that respond to pathogens in a generic, non-specific way. And then we have the adaptive immune system, which is a targeted and specific response. And so today we're going to talk about how do we recognize these pathogens, which is through the recognition of what are known as antigens. We're going to talk about the different kinds of immunity that you can get for the immune system and how that immune system is formed within your body. We're going to talk about the different types of immunity, like cellular immunity and humoral immunity, where one is looking at abnormally functioning cells, like with the cell-mediated immunity, and humoral immunity is looking at the fluids of the body. We're going to talk about how antibodies have different functions depending on what they're called, their class or their isotype. And then we're going to finish off with talking about once a infection is started, how the body responds, how these antibodies are mounted, how they're expressed and target and neutralize these pathogens. We're going to talk about the profile of how these antibodies occur when you first experience a pathogen for the first time, comparatively to when you experience a pathogen for a second time. And then we're going to talk a little bit about the vaccines that are required for attending school. So we talked about how there's those first line of defenses, which are those ever-present barriers like mucus and cilia, and salt that help prevent bacteria from being where it's not supposed to. We have those second line of defenses like inflammation and phagocytosis that responds to pathogens in a very generic way. Any pathogens can just trigger these responses. Then we have the third line of defense, which is very specific and targeted for one specific pathogen. And it does this using antibodies or T cell receptors in B cells and T cells. And this third line of defense is specific and targeted, but also we can remember what we've encountered in the past so that we can make better responses for that same pathogen in the future. We consider this first and second line of defense a part of the innate immune system. This is the thing that is given to you with you at birth, while the acquired immune system is not present at birth, but is gained through past and current infection histories. Whereas you encounter more pathogens, you can acquire these responses to those pathogens, and then you can remember those and memory for future infections. And so with that, we're going to start into chapter 15, where we're going to talk about the adaptive immune system, the cells that are involved in it, and how it gives us the immunity. We have, I'm sorry, I'm not actually in a presentation mode, so I apologize about that. But we talked about how the adaptive immune system has this specificity that's targeted to a specific pathogen, but then it also has this memories that we can remember this response that we have for that pathogen so that we can have this long-term response for that pathogen anytime we encounter it in the future. And we do this by recognizing what are known as antigens. And antigens are specific molecules or parts of a molecule that we can bind to and we can recognize as non-self. And so antigens stimulate the immune system by being able to be recognized by these receptors called antibodies or T cell receptors. And these, so you'll have a foreign material and then you'll have a receptor and it can bind to it. And it binds to the specific spot. The whole thing is called the antigen, but that specific spot is called an epitope. And so you can have an epitope here, and you can have an epitope here, or an epitope there. Any place that can bind could be a potential epitope, kind of like And this receptor has a binding site that binds to this epitope. And depending on that receptor's shape, it might be shaped in one way or it will be shaped in a different way. And depending on its shape, it will be able to bind to different epitopes or different antigens altogether. And so there are specific things like antigens have to be specific sizes, but also you have to think about like physically, the bigger a molecule is, the more spots there are to bind to. And so the larger or more complex an item is, the more antigenic it's likely to be. So like if you're looking at just a protein, you just have that protein surface. But if you're looking at an organism, you have that organism's entire cell surface and all of the proteins that are on its surface to target. And so it's something to think about when it comes to like what is an antigen and what's not, and like what we target, because like it's not just that we can recognize these pathogens because we just know that they're there. We're recognizing something very physically, and so we have to have a receptor that can physically bind to those, and we have these processes that take these receptors and can randomize them so that we can recognize these different epitopes and antigens. And so antigens can be all different kinds of biological molecules, and it can be non-biological molecules too, but for the most part, it's biological molecules. You have proteins and polypeptides, you could have lipids, nucleic acids, polysaccharides. Polysaccharides are always on the surface of like bacteria, and so that's always one that's important. Proteins are also are on the surface of different pathogens, and so that's always a target. And so these antigens can be categorized in a few different ways. So antigen is just like umbrella term. It is a substance that can be recognized by the immune system, right? And so an antigen can be recognized by these receptors. There are specific categories of these antigens that like just can be that can just be like just are special categories that they just have a little bit of nuance. So they have a special label. They're called alloantigens, and these are antigens or proteins that the immune system can recognize. But typically, the immune system is recognizing self and non-self. But what if that self is found in one person but is not found in another? And so with tissue transplantation, we have to make sure that the proteins that are found within a specific organ is going to be able to translate and transplant into a different person without that person's immune system recognizing that tissue as non-self. And so we have to go through the process of looking for what are known as alloantigens to make sure that they are a match and compatible with one another, to make sure that they are not going to recognize that foreign tissue as non-self and elicit an immune response. And so alloantigens are these antigens that can be found in one person or a different person, but they kind of that's a bad way of saying it. Alloantigens are these substances that can be found in a species, but are not always found in one individual of that species, right? So species is like a big thing, and then individuals are unique. And sometimes alloantigens are specific to this group and not that group. And so it can cause like transplantation errors and things like that. Anyway, we also have super antigens, and super antigens are these antigens that can elicit a very strong immune response within T cells and create these really strong inflammatory signals, and it can cause, just like with LPS, in causing like strong inflammatory responses. You can get tissue damage and shock and things like that from these super antigens. You have allergens, which are specific antigens that are that we haven't talked about IgE antibodies, but allergens are specifically antigens that elicit an allergic response, and that allergic response is caused by the recognition of that antigen by an antibody that uses what is known as an IgE isotype or class. And we'll talk about what that means later in the video. And then you have autoantigens, and these are things that that are like when you think of autoimmunity and like recognizing yourself improperly, that would be what an autoantigen is these are molecules that the immune system can recognize that it shouldn't actually recognize because it's something that's native to yourself. And so those would land into like tissue, causing tissue damage in autoimmunity. And again, the things that give us this, the adaptive immune system, its power is that specificity of being able to tailor a receptor to antigens, as well as being able to remember that receptor for later. And so it's that specificity and that memory that gives these antibodies, T cells, these antibodies, which are B cells, and T cell receptors and T cells, their power of the immune system. And so you have T cells, which are, yeah, I didn't even know that we were going to talk about this. We have T cells, which are T lymphocytes, which give us our cell-mediated immunity. And these ones have T cell receptors. And we have our B cells, which are B lymphocytes. And those give us humoral immunity. And those use antibodies. So antibodies are Y-shaped molecules, and T cell receptors are kind of just like regular receptors that are like just little sticks. But T cells give us these cell-mediated immunity. It's protecting and surveilling for abnormally functioning cells within the body to detect and eliminate any abnormally functioning cells like tumors, cancers, or intracellular cells that have been infected by intracellular parasites. Then you have B cells, and B cells create antibodies, which are those Y-shaped receptors, and they give us what is known as humoral immunity. And humoral immunity is like, if you think of like way back when, you're like balancing your humors. Those are like the body's fluids. And so humoral immunity is giving the immunity to the fluids of our body. So in between our cells, inside of our blood, inside of the lymph system, the antibodies are present and around. Our mucus even have different antibodies in them. And those antibodies are produced by different kinds of B cells and B lymphocytes. So cell-mediated immunity is mediated by T cells, and it's surveilling for abnormally functioning cells. And it does this through T cell receptors that recognize an antigen, and then it can have some kind of effect on the cell that it is surveilling. And sometimes this is looking for intracellular abnormalities. So a bacteria or a virus can get inside of the cell, and that cell will start to produce the wrong kinds of proteins. It'll have the wrong transcription profile. So essentially it's transcribing the wrong genes in at the wrong times. Maybe it's putting the wrong receptors on its surface. Maybe it's just like secreting out the wrong things. Something is causing it to be weird. The T cell is going to be able to help detect that and try to eliminate that cell, either by killing that cell or recruiting other cells around to eliminate that cell. And so viruses always have to go inside the cell to replicate. And so viruses are obligate intracellular parasites. You have bacteria. Some bacteria have to be in the cell to survive. And so those are also obligate intracellular parasites. And so again, those are where those T cells are helpful. And then with cancer and tumor development, T cells are defending against that. On the other hand, we have humoral immunity, and humoral immunity is given us by T cells, which produce antibodies. Again, antibodies are those Y-shaped proteins. And then they have an antigen binding region on either side of those. It's the same one, but they have two binding sites. Those T cell receptors have an antigen binding site right at the top. And so the antigen would sit right there and it would bind in there would be a binding site. The antigen is that substrate and it binds. Antibody, the substrate binds, or antigen binds right here. And so when that binds, it can do a couple of things, and it can help fight off those, and it can fight off those. It can alert the body to the presence of that pathogen, or it can help like neutralize and kill that pathogen. And we'll talk about like what functions those antibodies can have in a bit. But these antibodies can be free floating within the fluids of the body, or they can be bound to the surface of B cells, and they can be looking for like surveilling your tissues, looking for, it's really hard here. I'll just have one. You can have your cell, you can have your antibody. I guess your antibody's Y shaped. So there we go. And it's looking through the environment, looking for that pathogen, and it can bind and it can recognize that. And then it can also just be free floating around to bind. Like it says, it can be dissolved or it can be bound. And so if there was literally so look, there's a picture of it. It can be bound like this as a cell surface receptor, or it can be secreted out as a free floating protein. See, I just needed to go one more slide. Me trying to give you hand shapes to discern. But anyway, the cells that are involved with the humoral immunity are B cells, while T cells are for cell-mediated immunity. Both of these come from hematopoietic stem cells within the bone marrow, and hematopoietic stem cells within the bone marrow, typically when you're in fetal development, they're in all of your bones. Your bone marrow is found in all of your bones, and well, if we're going to start at the very beginning, in fetal development, your spleen is creating your blood. And then after birth, your bone marrow starts creating your blood components, your white blood cells, your red blood cells, your platelets. And then as you age, the bone marrow recedes from most of your bones and retains to just a couple of your bones, like your sternum, your ribs, your femurs, those large bones. Some people say long bones. Some people have said flat bones, but like, I don't know, your sternum, your ribs, and your femur at least, you know? And so these have bone marrow, and these bone marrows have stem cells called hemopoietic stem cells. And hemopoietic stem cells generate the myeloid lineage, which is the innate immune system cell lineage, the white blood cells that are for the innate immune system. And there is the lymphoid lineage. And you can think of the lymphoid lineage as lymph node or lymphoid tissue. And so that's kind of how you can differentiate the two. And so the lymphoid lineage can be turned into the B cells or T cells. And B cells develop within the bone marrow, and they then make their antibodies using what's known as VDJ recombination, which we won't really talk about too much, but they're essentially randomly assembling this antigen receptor site so that it can bind to a different receptor, a different antigen. And so one B cell will make one receptor binding site, and another B cell will make a different binding site. And that's how we can bind to different epitopes and different antigens all together, is because we can make nearly countless of these different binding sites to bind to nearly any substance or nearly any antigen, right? So anyway, B cells come from the bone marrow, and they develop in the bone marrow. Once they develop in the bone marrow, they can leave into the circulatory system, into this lymphatic system, and circulate and surveil the body for foreign material. You have another split of a different cell type, which are called T cells, which they leave the bone marrow. And there's actually a chemical gradient that they respond to using Notch signaling. Essentially, you know, how we talked about how bacteria can move towards or away from a stimulus using chemotaxis. Essentially, chemotaxis is what's happening, right? So this cell has a receptor that can recognize this compound that's being made in the thymus. And so when the cells leave the bone marrow, they travel into the thymus, where they develop into developing T cells. they undergo the uh, developmental checkpoints and that VDJ recombination that randomizes that binding site. So now the B cells have a randomized receptor and the T cells have a randomized receptor. Then the T cells are in the lymph node already, and they can then surveil the body for abnormally functioning cells. Again, those B cells look for pathogens just free floating in the body, while those T cells are looking for pathogens that are inside the cells or abnormally functioning cells. We have two different kinds of T cells. Typically, you have cytotoxic T cells or helper T cells. The cytotoxic T cells are essentially saying, "Oh, you are weird. You are not acting right," and it's going to tell that cell to go and to undergo apoptosis, which is essentially the process of cell death. It is one of the processes of cell death. There's a couple of processes that you could choose. And then there's helper T cells, and helper T cells recruit more immune cells to the site. And they're like, something's going on here, and we need to figure it out. Okay. So then come all over here and try to recruit all of these immune cells to the site and try to figure out what's going on. Is there like a tumor? Is there an intracellular pathogen? Or is there a pathogen that has been digested and been presented to T cells for this process to occur? And we'll talk about what that means in a second. You then have two different types of B cells. The B cells have memory B cells or they have plasma cells. And memory B cells are the ones that we remember the responses for later. So like we make this receptor. Once we were like, oh, that was a good receptor. We can remember it for later, and we remember it by making these memory B cells that are essentially going to be like an archive of these receptors for us. And then we have plasma cells, which are these secretory cells that secrete antibodies out of themselves into the interstitial fluid and into the body. And so this is essentially a factory to produce these antibodies to recognize and neutralize the pathogen. So this is going to be what we use like at the height of infection, of like trying to get a hold and like neutralize the pathogen from like continuing. So this is a description of that figure. So I'm going to skip that. And so again, we have T cells, which are giving us cell-mediated immunity, which is looking for abnormally functioning cells. And it does this through what is known as the major histocompatibility complex. And these are also part of the like tissue rejection stuff, I believe. But what is happening is these histocompatibility complexes, or MHCs, are essentially like an auditor of the cell. Every cell has them. Some of them have one form and some have another, but either way, they're like an auditor of the internal state of the cell. So you have your cell, and there's proteins, enzymes, and things. Sometimes you might bring bacteria inside of it and it might get cut up. Sometimes you'll have your proteins and they'll be working. And so MHC, what happens is that all of those innards, some of them get broken up into pieces. They bind to this MHC, and the MHC is put out onto the cell surface of the cell. And this is what is called as antigen presentation. And it's essentially saying, this is what's happening inside of me. Please look at it and tell me if it's normal or not. And a T cell will come and it'll look at that MHC complex. The MHC complex and that TCR will bind where the antigen is sitting right in the middle. And I don't have enough fingers. That's close. And that antigen, where my thumb is, this one, is being recognized by both of these receptors. And only when that happens can the T cell recognize this antigen and say, okay, you're normal or you're not. And if you're not normal, we're going to have some kind of activation. Or is that going to be recruiting other immune cells, or is that going to be telling the cell to undergo cell death? And so we have helper T cells, which are helping to recruit these cells to the site of infection. They're going to be increasing like inflammatory responses. They're going to be poisoning the microenvironment within that space for the immune system to be better situated to attack the pathogens. You have cytotoxic cells, which are secreting out enzymes that specifically kill cells or tell the cell to die. And then you have natural killer cells, which work at specifically targeting cancer cells. These cytotoxic T cells also look and target cancer cells. You then also have beta delta T cells, which are found in like your epithelium, but that's kind of in the weeds. But they are kind of like a bridge between your innate and the adaptive immune system. And they can recognize like uh pathogen they can recognize like antigens within your blood, and they can recognize antigens within the like epithelial layer of your organs and the mucus layers as well. But that's like in the weeds. You really just need to know CD4 and CD8, where CD4 is what is known as a helper T cell, and CD8 is what is known as a cytotoxic T cell. And so what happens is, again, we have these cells, and they have these MHC complexes. And depending on the cell type, you'll have an MHC2 complex or you'll have an MHC1 complex. All of your nucleated cells have an MHC1 complex, while only your phagocyte, what is known as a professional antigen presenting cell, which are these like macrophages that are, and like neutrophils, which are professional at phagocytosis, where they consume and swallow up pathogens inside of them, cut them up, and then put them onto their cell surface. And these professional antigen presenting cells are typically a different cell that's a part of the immune system. While MHC1 is all nucleated cells, this is just like your regular tissue cells. And so we have some layer of surveillance from both parts of your immune system, and we can have surveillance from your just regular tissues. So because this professional antigen presenting cell is like a macrophage and it's a part of the immune system and it's helping to clear out infections, we really don't want to try to kill it, because it's most likely not being an issue. It's probably helping us and telling us that there's an infection. And so what we use is these T helper cells, which are CD4 positive. CD4 positive is these clusters of differentiation. All they are these different kind of cell surface receptor. And this CD4, you can see depicted here, complexes with the T cell receptor, the MHC complex, and the CD4 all together with the antigen. And together, this activates the CD4 T helper cell. And again, kind of how I remember it is that this CD4 recognizes this MHC2, and that MHC2, I guess you can remember, okay, this is a couple of steps to remember, right? Professional antigen presenting cells, which are like a part of the immune system, they are professional, and so they have a higher number for the MHC2 complex, right? You can think of like when you have a job, you're like data scientist 1, data scientist 2, data scientist 3, senior, whatever. You have your everyday normies, MHC1. You got your professionals, MHC2, right? We don't want to kill our MHC2, so we're going to use our T helper cells to help them instead of kill them, right? But all the normies we can kill. This is where the analogy breaks down, right? All the normies we can kill with our cytotoxic T cells, which are CD8. So I guess that's where the analogy breaks down. But either way, our CD4 is helping this professional antigen presenting cell. And it can do so by essentially changing that microenvironment by releasing signaling molecules, cytokines, interferon gamma, things that uh, cause inflammation or recruit other immune cells to come to the site of infection. Then you have the cytotoxic T cell. This cytotoxic T cell is looking at the regular tissues within your cell. These regular cells could have a pathogen inside of them. We wouldn't want that, so we'd want to kill that cell. It's not helping us with the immune system, so we want to, we don't expect them to be just like, hey, I swallowed this pathogen just to be helpful, so like here you go, like the macrophage did. Instead, we're thinking that maybe they were infected with a pathogen, and just by chance, that cell was able to digest it and put it onto the cell surface for auditing. And so this also can be for like cancer cells, right? And so if you have these abnormal proteins, those abnormal proteins are presented out in their MHC1 complex, and these cytotoxic T cells recognize that MHC1, and they're like, oh, that's not normal, and you're probably not a part of the immune system, so we're going to release these enzymes that will eventually kill you, where it'll either tell the cell to start the cell process of cell death, where it starts to like put up apoptotic factors. Apoptotic factors are proteins and enzymes and things that start the process of apoptosis. Apoptosis is the process of like, you got a cell and that's apoptosis. If you don't believe me, you can look up a video, and it's essentially the exact same thing. Truly, you can just trust me. Okay, it's a little bit different, but it's kind of the same thing. It's essentially the membrane starts to break down, the membrane starts to bleb. It's called blebbing. They start to make these little bubbles, and they kind of just disintegrate open, and it lyses the cell open. And you can kind of see how that kind of cracks and things, but in real life, it looks like little bubbles coming off of it. And so, yeah, so you got these T cells, and they're surveilling the body in different ways, where it's sometimes looking towards the immune system to help their immune system by boosting them, by helping them with CD4 helper T cells. Other times it's surveilling the body, looking for cancer and abnormal functioning cells with cytotoxic CD8 cells. But either way, these T cells are a very prominent portion of the adaptive immune system, and they can regulate the microenvironment in quite a big way. And we talked about how super antigens have an overactivation of these T cells, and essentially it's causing them to do all of those functions just a lot. And so this is going to cause, what's that going to cause, right? You're going to have so many of these cytokines and interferons and inflammatory processes being increased, and macrophages do phagocytic behavior. This is going to cause like large inflammation, which large inflammation when it's like a lot of it can cause tissue damage. You have cytotoxic effects, and if we're just creating out cytotoxic signals really fast and at a high pace, that's going to cause tissue damage. And so you can cause high levels of inflammation, you can cause toxic shock, you can cause tissue damage or organ damage. And so it can be quite a bit of an issue. And depending on the antigen, if the super antigen is localized, then like maybe those effects are going to be localized. But what if that super antigen is like able to get into the circulatory and get all over your body, right? We're going to have those kinds of inflammatory responses happening everywhere, right? And so I don't have an example of that, but like thinking of what could happen with the different kinds of these super antigens is really important, and how can they interact with the different systems that you know about. So then we have humoral immunity, and humoral immunity uses antibodies. Remember, antibodies are those Y-shaped proteins with binding sites on either point of that Y. And they can recognize a pathogen by recognizing an antigen, a specific place on that antigen is called an epitope, but it binds with that antibody, and it can cause a few things, but one of them that we're going to talk about is going to cause phagocytosis, and it's going to allow for that bacteria to be eaten up, consumed up, and then presented out. Remember, we talked about how this cell, how essentially the same process. We just talked about this. We can eat it up and we can go through antigen presentation because this cell is a part of the immune system and it is professional at this. It's going to have MHC2 because it has a higher number. And MHC2 is going to be able to complex with T cells. And so there's an interplay between the humoral immunity and the cell-mediated immunity between your B cells and your T cells through these complexes even. And so this is going to, this could stimulate your CD4 T helper cells. And what would that do? That would increase different cytokines and interferons and increase the inflammatory response. And it's going to activate this B cell. And once we have an activated B cell, it can either be turned into a memory cell that we can remember that receptor for later, or we can turn into a plasma cell. And these plasma cells are these factories for antibodies that are being secreted out to neutralize and recognize a pathogen. Again, this is a description of that figure. So again, you have this Y-shaped protein, and this Y-shaped protein is a polypeptide chain, right? Because proteins are polypeptide chains. There are 2 unique chains, but there are 4 total. You have this light chain, which is the shorter of the 2. You have this light chain. Those are the same chain. They're just dimers. There's two of them together. Then you have this long chain, and this long chain is called a heavy chain. And there are 2 regions here that come together as a binding site. And that's where your antigen comes in and sits down and binds. And you can see how antigens, oh my goodness, that scared me. A napkin flew across the doorway, and it was like, I don't know, a squirrel was gonna come and eat me or something. I don't know. Anyway, these antigens have this binding site, and that binding site, depending on its shape, determines what antigen it can recognize. And we can randomize this. We can essentially make any binding site for nearly any antigen using the process of VDJ recombination. But this antibody can be broken down not just into chains, but also into the Fab region, which has this antigen binding site. And this is called the antigen binding fraction, but they put the F in the front so that it makes sense with this one, which is the crystallizable fraction, or the FC region. What does this mean? The FC region is crystallizable. And so there's this process in structural biology, essentially how we study proteins and the structure of proteins. Apparently, you can take a protein and you can, because it's so orderly and folded just right, you can make it crystallize. And crystals are just repeating units of a single it's a lattice of a repeating unit in a lattice formation, right? And so you can think of like any crystal, like a diamond has hexagon, like has those carbons in those carbon rings, and they are layered in this crystallizable lattice. Proteins can just be like, here's a protein, here's a protein, here's a protein, and they can be so compact and they can be so ordered that you make a crystal out of it. And you can shoot lasers at it at really high energies, and you can just be like, look at the light that reflected off of it. Well, is essentially looking at like the opposite of shadows, but you're looking at like the shadows essentially that it creates. And it's the light spots and it's how the light beam is refracted off of the crystal, and it's called diffraction or whatever. But like in the weeds, you don't need to know this. This is just like some cool pieces of information, right? So we're shooting this laser at this crystal. Because of how the crystal, how these proteins are packed and how the folds of the protein are, causes this light to be bent and reflected in a certain way. And we can do some really complex math to map that back to the structure. And we can essentially say, this protein is folded this particular way. So this region here, very early back when we're first studying these antibodies, was really easy to crystallize because it's the same fraction, it's the same sequence, it's the same structure. So it's easy to crystallize. This region, because it's variable in every antigen, each antibody has a different region because it recognizes each receptor recognizes a different antigen. It was very hard to get that to crystallize because each of them have different folds. And the whole thing about this crystal is that it's like repeating units of the same structure over and over and over again. And so because these are all kind of just a little bit different, made it like the equivalent of it made it blurry, like it just didn't really work. And so that's where those names really came from, just so you like kind of understand where they came from, but it's not really necessary for you to know. The most important thing I want you to know is where that antigen binding region is, that it's varied, and it would be nice to know the structure, but it's not like super important, but like it's pretty standard to understand. So antibodies can have a few different structures and a few different functions. You can have what is known as complement fixation, where we talked about in one of our last videos, where the complement system is this process of building a complex, initiating a cascade of protein reactions that build a giant complex on a bacteria's surface, and it punches a hole through the membrane of that bacteria, and it lyses the cell and causes the cytoplasm to leak out of the cell. And so the antibody is one of the things that initiates that process. And so the antibody can initiate fixation of bacteria because it has two binding sites. One binding site could recognize two different, one binding site could bind to one bacteria while another binds to another bacteria. And this can create this like network or cluster or aggregate or clump of pathogens that's going to be easier to recognize and target for phagocytosis. And they're going to be maybe easier to clear from the body. But essentially, these pathogens are going to have less function because these antigens are what is doing agglutination, which is causing these pathogens to aggregate or agglutinate. And then you have opsonization, where the antibody coats the bacteria, and because there is essentially a binding site for macrophages and other phagocytes on the stem of that antibody, that macrophage can recognize that way more readily than the pathogen itself. And essentially, it's like a handle for it to grab onto for phagocytosis. And so this opsonization process makes this phagocytotic process much more reliable. You have neutralization, where typically you have a toxin or something that has a specific binding site or something that's crucial for function, like a HIV has a specific receptor that it wants to bind to get into the cell for replication. If your antibody is able to block that receptor binding, that virus can't get into the cell. And so this is what is known as neutralization, is that we're blocking fundamental biological processes using these antibodies by coating them either in the antibody and physically blocking them. And this is going to be what is known as neutralization. Similarly to precipitation and similar to agglutination, precipitation is the same kind of idea that these antibodies are able to bind to a toxin in a very succinct way that causes them to coagulate together, clump up, cause aggregates to form, whatever you want to call it, or cause a precipitate to form, which essentially is causing, there's definitions of that about like water being pushed out, where like water is pushed out of like the complex and it's causing it to fall out of solution or whatever, but like it doesn't matter. All it means is that it's causing it to clump up. That's going to make it where maybe that toxin isn't going to be active. And that would be neutralization. Maybe it's just causing it to clump up, and that's going to be easier for it to be targeted for phagocytosis. And then you also have toxins that can be neutralized as well using the same kind of process. And so we talked about how the we talked about how the crystallizable fragments are have binding sites where these phagocytes, like macrophages, neutrophils, all of these cells that are able to do phagocytosis, or like mast cells that are doing important signaling, they can bind to that receptor, and it's like a handle where they can say, oh, you are indeed bound to a pathogen. We need to either swallow you up and do digestion, like with macrophages and neutrophils or eosinophils, or undergo signaling to remodel the microenvironment, recruit more immune cells, like with the mast cell. And so the complement system, we talked about how the complement system builds up this complex that has a hole in the membrane. And this is all facilitated by a antibody that first recognizes the bacteria. And this causes what is known as the classical pathway. There are a couple of pathways that can activate the complement system, or like alternative complement systems, but they have the same kind of idea or the same kind of flow. And so the idea is that the antibody recognizes this pathogen, and then it converts the C3 protein. I'm going to zoom in real quick. So the antibody recognizes the pathogen, and this triggers this C3 convertase to break the C3 into two parts, where there's the C3b and the C3a. C3a is going to go over and do signaling and help with the immune system, like boosting the immune signaling, and while C3b is going to go to C5, and C5 is going to get cut in half. And C5a is going to go and do signaling and help the immune system, while C35b is going to go off, and a set of complexes are going to come together around C35b. C6, C7, C8 all come around C35b to form this complex. And then multiple subunits of C9 all form around this structure to create the pore. And this pore is known as the MAC complex, or the membrane attack complex, MAC. This creates a pore where the cytosolic fluids are able to escape, and it causes a lysis of the cell. Now, what's important about this is a very complicated this is a very complicated pathway, and you don't really need to know all of the components or steps of it. if you want to know it. It's really cool. It's really important. It's really interesting, and it has some important implications in biology. Um, excuse me. And I would encourage you to watch a animation on the classical pathway and the complement system, because it's really cool how these how these proteins come together to form this complex. But what's important is that this is a cascade of one enzyme working on another, working on another, working on another, right? So this enzyme has a substrate and a product. And that product is an enzyme that has a substrate and a product. And that product is the foundation for a complex. This complex makes that MAC, right? And so what's cool is, because you'll see this in a lot of systems in biology, where there's this signaling, there's a pathway or like these steps. What's cool about it is that we have each of these steps, we have the potential of this C3b enzyme can then act on not just this one C5, but maybe 10 C5s or 20 C5s. And those C5s are going to be made into 20 C5bs. Just pulling out numbers from my ass, right? And these C5bs could maybe make 5 to 20 complexes. And so from this, just this one C3 reaction, we're able to make 20 complexes, these MAC complexes. And so what you can take from this is that with each of these steps, the signal can be amplified essentially. So one of these enzymes can activate 5 more enzymes, and that enzyme can activate 25 more enzymes, and that 25 more enzymes activate so many more enzymes. And so this is how we can take one single signal from that one antibody and turn it into many of these pores that are going to lyse and kill the bacterial cell, right? Okay, sorry. And then we talked about how these immunoglobulins have different stems, or what are known as classes or isotypes. You have IgG, IgA, IgM, IgD, and IgE. And I remember this by MAGE, MAGED, but you could also remember this by GAMED, GAMED. Either way that you remember what ones exist, each of them have their own function and are used in different ways, and they have different structures as well. They all have that Y fundamental structure, where you have those light chain and heavy chain, and you have that heavy chain that has this constant region. And this constant region, or this C region, is what is varied between these isotypes. And so IgG and IgM are those typical ones that you're going to see within your B cell immune system. This is IgM is the first kind of antibody that's produced. These are low specificity, but they they're the first that they're produced, and then they are refined, and they have like a secondary randomization. And then you have IgGs, which come up second after a pathogen exposure. And these ones are going to be much more specific in what they can target. And so these ones are kind of loose with what they can target, and these are a little bit more specific. And IgG and IgM are both used inside your B cells for, not inside, but like used by your B cells inside of your body's like blood circulation and inside your tissues to fight pathogens. IgA is something that's found in your mucosal layers. So inside your respiratory tracts, inside your gut, these antibodies are secreted into the mucus to recognize and neutralize pathogens. We also have IgD and IgE. IgE is what's important in allergies. And so this is going to be what activates mast cells, and it's going to give us allergic responses. And then IgD is used in respiratory, found in the respiratory mucosa, but it is still trying to be like its complete functions are still being kind of worked out. And so one thing to mention is that when we talk about what these constant regions are binding to, this is like, remember IgG is going to like opsonization, can coat like a pathogen. And then the phagocyte is going to, which could be like a macrophage, it could be a neutrophil, it could be a dendritic cell. Those are going to recognize this constant region, and that's what it means when it says it's going to bind to. Same with the epithelial cells bind to the constant region of the IgA. And mast cells, which are those signaling molecules, recognize the IgE's constant region, which is why they're really important with the allergy responses. They're going to degranulate and cause that, release histamines and inflammatory markers that are going to cause that allergy. So we're gonna, I think we kind of covered all of this. Like some of these things are important to know about, like their lifespan is nice to know because IgG are those like specific long-term um antigens. They are those more specific receptors for pathogens. They last a little bit longer in the blood. And so when you like have an infection, you could even see that you had that infection even up to 20 days after you had that infection. We talked about how the bind to is talking about the FC region or that constant region. And then we can talk about how when you have a bacteria and it gets into the body, it creates an immune response. And that immune response typically is those second lines of defenses, those innate immune responses. But if that innate immune system can't get a hold of that infection and that infection continues, your adaptive immune system is going to start to be activated. And we're going to be looking for a receptor that can bind to that pathogen. And this takes quite a bit of time, 7 to 14 days, until we can recognize and select a receptor that we can recognize this pathogen. And the first receptors that we can select are IgMs. And this is on our first exposure to a pathogen. So we've never seen this pathogen before. So we're essentially starting from scratch. After we find receptors that are kind of working, remember IgMs have kind of low specificity. We find ones that are kind of working and they undergo somatic hypermutation, which are essentially they're going to take that receptor binding space and they're going to say, well, let's just tweak this here and tweak that there, and let's do this and let's do that. And now we have hopefully, a better antigen binding site. And this process also converts it's not the same process. You have that somatic hypermutation, and then you also have class switching that happens. And that is where we get the IgGs. And these are going to be those higher specificity antibodies that are recognizing that antigen at much higher like levels of affinity or much better. And so they're going to have a better response, and they're going to be much more effective at fighting the immune system. So sorry. And eventually the infection will be cleared, hopefully. And these antibodies levels are going to go down, and some of them are going to go into memory. So then we're going to encounter this pathogen again in what is known as the secondary response. That first response was called the primary response. The secondary response, we've seen this pathogen before. We just have to remember if we've seen it before. And so we have to go into our memory, essentially. I'm kind of talking in this like kind of hyperbolically, we don't go into like a memory and like look for it, but kind of, so essentially what's happening is you have a pathogen in your skin or in your tissue. You have your innate immune system that's fighting that infection, that pathogen. And it's going to, those immune cells are going to swallow up the pathogen, chew it up, and put it onto its cell surface through antigen presentation. And some of those are going to be looked at by B cells or T cells, and that's going to essentially communicate the innate immune system to the adaptive immune system. And that communication there is going to be like, we're going to be looking for a receptor that can bind to that pathogen. Excuse me. And so we're going to have an antigen that our let's say our macrophage, right? Our macrophage or a dendritic cell has a pathogen. It's chewed it up, put it on its cell surface. We have it on our cell surface, and then we're going to be looking for B cells that can recognize it, or we're going to look for T cells that can recognize it. And we're going to find a B cell, an IgM, or an IgG that we've used before, that we've developed in this primary response. And then we're going to be able to take that cell and say, okay, you grow and develop through multiple cell divisions, and we're going to make lots of your cells. We're going to make plasma cells that secrete out antibodies to fight this infection. And then we're going to store some for memory for later, just in case, just in case. And so what's important about this is that we have, because we've took this time to develop a response, and then we can remember that response for later, we're able to mount a response, a secondary response, so much faster than when we would have beforehand, right? We were able to, we were exposed to the antigen way early on in both cases, but we didn't have a response. We didn't see any antibodies in the blood until 15 days later on the primary response, or like 3 days after a secondary response. And so this is where the power of that memory comes in, because we can remember those specificities and those receptors that have specificity. We can like kickstart our immunity for infections that we've seen before. And so we can have what is known as active immunity, which is what we've been talking about, is that we've seen an antigen, we've looked for a receptor, we've proliferated that cell, made many of those receptors, we've tweaked that receptor, and then we put it into memory for later storage because we know it works. This is called active memory. And this is, we can access it anytime, just like any other, just like what we've been talking about, right? Passive immunity is like, if I've given you antibodies and you can't make those antibodies or those receptors yourself, this is what is known as passive immunity. This is like an immunotherapy. If I inject antibodies into you, eventually those antibodies, like any protein, it's going to be degraded within your body over time. And this is going to give you a short-term effect, and you can't create those antibodies, and so it's short. We don't have a memory with that. And this is just what we find with like our medicines that we perform with these antibodies. Then you have natural immunity, which is the ones that you acquire through natural exposures through your day-to-day life. Then you have artificial immunity, and these are the immunities that you get through vaccinations. These are things that we introduce an antigen that you can mount a response to. And this kind of gets into what vaccinations are. And this kind of gives you how you can have combinations of active and natural and things like that, where you can, but I'm going to kind of skip that for now, just because for the sake of time. Here's another example where you can have active immunity, where you're getting natural active immunity, where you're just getting sick. You're getting natural passive immunity from getting what antibodies is it? IgG antibodies pass through the placenta, and I think also through breast milk. And so passive immunity can be given through mother through child, or it can be given through medicine, which would be that artificial. And then you have that artificial active, which would be like vaccinations. Vaccinations, yay. And so we talked about that primary response. We talked about that secondary response. Vaccinations are a way of kind of skipping that primary response. We're essentially giving this first exposure without having the detrimental symptoms that are associated with having an active infection. You're not going to have the tissue damage. You're not going to have the inflammatory, like, you're not going to have the like the over inflammatory response, like beyond just like regular, I got an injection and it's going to make things like red, right? You're not going to have, I think the tissue damage is like a big one, right? But you're not going to have like your entire innate immune system being like, we must fight this pathogen. We're essentially specifically trying to target the adaptive immune system, targeting the B cells and the T cells to create these receptors so that when we actually get an infection that's real and actually have symptoms, we don't have to wait 15 days to get a response to be able to treat it ourselves. We can make our own antibodies to help ourselves fight this infection within days instead of weeks. And so this is where vaccines are good. This is where the power of vaccines come from, is because you can, instead of being miserable and risking your life for this amount of time, you can skip it. But there's things to be to take into consideration, right? And so again, where you have this principle of skipping the primary immune system and getting that second to skip to that secondary immune system. But the things that you have to remember is that you want your immune system to be able to stimulate your B cells and T cells. You want it to give us lasting immunity. You want it to not be toxic to your cells, not give us tissue damage. You really don't want it to elicit your innate immune system too much. And you want it to be able to be practical in your day-to-day life where you're not always getting vaccines. We know that flu vaccines, just because they're harder, they're annual, they're harder to get. And so there's barriers to treatment. And so that's not ideal. Sometimes that's required, right? We'll learn this in like, I think unit 6. There's so much diversity within viruses and within the flu strains that they have to have with current vaccine technology. They have to have an annual flu vaccine. But ideally, you would just have one vaccine and be done completely for the rest of your life. And you would want it to be able to be inexpensive and be able to be available to all people, regardless of their income or location. And you want it to be able to be self-stable. And that's important with like location, right? Because some places don't have the same kinds of storage mechanisms that other people do. And so we can make vaccines in a few different ways, and it's come a long way. It used to be where we were using just make them sick, you know, chickenpox parties, make everyone sick. And we are going to be essentially vaccinating our kids. We're planning a specific time for them to get sick. We're going to take care of them, and then they'll be fine later on. However, you're making them sick with like the actual thing. And so you have literally this exact thing that happens. You're just going to be prepared for it, right? It's not going to come as a surprise. We've come a long way from that. And we've come a long way from like giving just like testing. I'm thinking of like how we took like pus from other patients and like put them, not when us, I didn't do this. This is like, centuries ago, doctor or whatever in some kind of ward or whatever, like scraped like pus from like a wound or whatever and like put it onto someone else, and like they were able to be protected from their like they were able to be protected from, so gross, be protected from their from infection. Thankfully we don't do that anymore, but what we do is we give them vaccines, and these vaccines can be made up of a few different things. They can be made up of entire bacterias that have been killed, or for viruses inactivated. Or we can give them weakened versions of those strains. You can think of like what was it? Chicken pox. No, there's smallpox and cowpox. You could give yourself cowpox and garner protection from smallpox. And so you could, even though it was a live cell, you could say that the cowpox was an attenuated or weakened version of a virus that was better than smallpox. But like I'm thinking that's kind of stretching the definition. Typically, what we do is we knock out virulence factors from a pathogen so that it's just not so deadly. Maybe it needs specific nutrient requirements now. Maybe it needs, maybe it doesn't have the enzymes that it usually has that it can like infiltrate the tissues as well or can attach as well, right? Anyway, I will talk about that more in a minute. And then we can also have specific molecules instead of the entire uh, pathogen. We can just have a piece of that pathogen. And again, we can either just chop up the pathogen itself and use that, or we can genetically modify organisms and create that piece of a pathogen synthetically. When I say synthetically, it's biologically, but like using genetic engineering. In silica, in vivo, not in silico. That would be in a computer. So you have vaccines that can be using killed and inactivated pathogens. This would be if you have a pathogen and you like heat fix it, or you put a chemical in it and it destroys it, but it still leaves all of its body parts and all of that all around. And so because it's the entire pathogen, it has all of those surface receptors and things that could be potential antigens or immune stimulants. And so this is one option. You could have an attenuated vaccine, and this attenuation process is essentially taking out those virulence factors and making it where it's less likely to be able to spread across people groups, or making it where it is less likely to cause serious illness. Now, I will say that there are some viruses, some vaccines, that one thing that they want it to do is for it to be infectious. So when you have a limited budget, or you have you can't vaccinate a whole population. If you're able to vaccinate maybe 10%, and those 10% are able to make some other people sick, maybe you can boost that immunity up to 20%, or even more. But because of what they're getting sick with is something that causes just a mild cough or like mild symptoms, it's not life-threatening. It's not going to be harmful as it would be if it's a whole virus, and it's going to allow you to vaccinate more individuals that way. However, there are problems. These are living things, and so mutations can happen and they can gain those virulence factors back. They can either, if you just like broke it with a mutation, those mutations can be reverted. If they're like a simple point mutation, usually you would be like completely cutting out a gene, so it's like really unlikely to happen. But also we know that bacteria and stuff take up whole things of bacteria and whole things of DNA through transfection, or they get uh transmitted from bacteria through conjugation. And so the genetic makeup of these pathogens, even if they're attenuated, can change. And it doesn't mean that they don't get less pathogenic. They can get more pathogenic. And so even if you have these attenuated vaccine strains, what if one becomes dangerous? Or what if someone has an autoimmune disease? Sorry, what if someone has an immune deficiency and they're just not able to fight off even the weakest strain, and their sickness is so much worse than anyone else's? So there's there's pros and cons to having these kinds of immune vaccines. Um, so then you can have something that's a lot safer is using just like a single piece because you don't have the genetic component, you don't have the entire bacteria. You just have this one piece that we know that is going to be good for the immune system to target. It's going to be a good antigen. Typically, they've done tests at this point to know which pieces to look at. And so you take your bacteria or your pathogen and you cut it up and you purify from culture that piece. This is great because it's exactly what it looks like. But you have to grow a ton of this pathogen to make this vial. And so it's a dangerous process for the people who are manufacturing this product because they are handling not only the pathogen, but also potentially the toxins that it includes. A much more safer option is to take the genes that make this antigen and put it into something much safer. And this is where genetic engineering and biotechnology comes from, where we're able to take an antigen and the gene that codes for that antigen, put it into a plasmid, which is that circle of DNA, has a promoter that causes it to express. We put that into like yeast or bacteria that's like E. coli, that's like something that's not super dangerous. And then it's going to express that protein. And it's going to do so in high numbers. And because like we've been culturing yeast for centuries, we can have giant bioreactors of yeast making this small protein or this antigen, and then we can purify it down from those giant bioreactors and use that and never touch this pathogen, never even have it in the lab. Because we have the sequence for it, we can generate that sequence without touching and purifying it from it. And so this is a much safer process, and it can be done at so much higher capacity, so much cheaper, and it's a lot more it's usually more stable. Now, one thing to remember is because we're not giving an entire pathogen, we don't have all of those different antigens to choose from for the immune system to make a response. And so while these vaccines typically are cheaper, are more stable, and are faster to produce and are safer or whatnot, there is a downside that you only have this one protein to bind to, right? Where are you going to bind to it? Just the surface. You're going to have to find somewhere to recognize and neutralize. If you have this entire if you have these entire pathogen in here, you could make an antigen for any part of it. And so just because of the size and the surface area, this has a much higher diversified response. This kind of vaccine where you have these kind of vaccines that have the entire organism stimulates the immune system to have a much more diversified response. While when you just have a piece, you have a much more limited range of receptors. So if you remember back with COVID, when someone got sick with COVID and like the actual virus, their immune system, they were able to create antibodies for not just the spike protein, but also the other accessory proteins that make up the capsid that surrounds the COVID virus. While people who had the COVID vaccine just had the spike protein. And so they're able to just make antibodies onto that spike protein. That spike protein is very important for the entry into the cell. And so it's biologically significant. and it's required for functioning. So if we can get an antibody to block that or bind to that receptor, it's going to maybe neutralize it where it can't get into the cell. But also another thing is that because it's so important, it's highly conserved across different variants of that COVID virus. And so if you have a receptor, an antibody that can recognize that highly conserved receptor, even if you have viruses that have adapted and changed, that receptor is likely to be similar or at least close enough for that antibody to still recognize. But if you recognize like some kind of protein at the bottom down here in the cell membrane, it could change a lot faster. And so there's always like, it's a very complex system, and you have to kind of think about it from different angles to understand kind of how these vaccines interact with the immune system. And like, they absolutely bypass that primary response and give you a secondary response. That's true. And then there's that second level of like, how are we creating that response? What is that response? How diverse is that response? Do we have one response only, or do we have 30 responses that we could choose from? What does that mean? Do we have one antibody that can bind essentially to one receptor, or do we have 30 different antibodies that can bind to maybe that receptor and this other receptor and this other surface protein and whatnot? Does that make sense? And so then you can have RNA vaccines, and RNA vaccines have these are these RNA molecules that can come into the cell and then can use your ribosomes to translate a protein that you wouldn't be able to make without this mRNA. This mRNA then gets degraded, and this protein is the antigen that we're wanting to make an immune response to. And so we have to inject the mRNA into the body has systems to detect and eliminate naked nucleic acids. So DNA and RNAs, we expect them to be in nucleuses, not just free floating. So we have enzymes that break them down within our body. And so we have to cover these molecules with a lipid nanoparticle to allow them to be injected into our body and like free float into our cell to get to our cells without being degraded. They merge with our cell membrane and release the vaccine into they release the mRNA into the cytoplasm. The mRNA goes to the ribosome. The ribosome goes to translation. We have this antigen. That antigen is going to get secreted out into the environment. That is then going to be picked up by the humoral immunity in B cells to get or phagocytes, and then T cell immunity with the cell mediated immunity. So we talked about how the RNA is translated into proteins. And so these vaccines are important for protecting those who have a much harder time fighting off infections. And those who aren't able to get vaccines. So vaccines for the individual helps to skip that primary response and have a much faster response to an infection that they've never had the actual pathogen for, skipping to that secondary infection response. But for a population as a whole, there's something called herd immunity, and that's kind of working cooperatively with everyone as your neighbors to kind of make sure that everyone has their best protection they could possibly have. And so in public health, there's this idea of herd immunity, is that as long as you have a high enough percentage of the population who is vaccinated or is immune from a particular pathogen, that pathogen is going to have a much harder time spreading across the population, especially within especially within individuals that aren't vaccinated or can't get vaccinated due to health reasons or through choice. And so we have to make sure that we have a high enough vaccination rate across the populace to effectively keep the surrounding to lower the risk of the spread of these pathogens. And we can kind of see how, as we lower our vaccine efficacy with, I mean, our vaccines rates, like within, for instance, like with measles, we can see how that herd immunity is starting to wane as we're getting more and more spread of that disease. And we can see that if we had more vaccinations and a higher percentage of individuals that were immune to it, it would kind of isolate the people that are infected, and they couldn't spread it to anyone that they're around because everyone that they're around is vaccinated and is immune. But it's unfortunate that we seem to be going in not a great way in this category. But it's really important to understand what herd immunity is, why it's good, not only for those who can't vaccinate themselves, right? So the elderly. There's some vaccines that aren't given to infants, so the infants and immunocompromised individuals, and those who couldn't do it for like religious reasons or just for choice, even if that choice I don't agree with. But it's important that if we had a higher herd immunity, those people would be able to make that choice with a lot less with a lot less of a with a lot lower of a risk. So these are the vaccines that are required for school-aged children and for daycare in the state of Oklahoma. We can have hepatitis V, hepatitis B, diphtheria, poliovirus, measles. Something that's really cool about these vaccines is Tdap is a vaccine that is one vaccine, but it can give protection for multiple pathogens. Same with the MMR vaccine. It gives us protection for multiple diseases, measles, mumps, and rubella. You also, I guess you have varicella that could protect you from chickenpox and from shingles, but both of them are varicella virus. And so with that is the units